Hello student, how are you? I am Naran Mohanty. Hope you all will fine. Okay. Today we will be discussing about the process replication of DNA. Are you getting my point? So this video duration will be 10 minutes around. Hope you all will enjoy the video. Let us start. Okay. See, DNA. In previous lecture we have already discussed what do you mean by DNA and what do you mean by transcription, all these things. Okay. Simply just remember, DNA means it is a polymer of the nucleotide. Are you getting my point? It is a polymer of nucleotide. Okay. That means many nucleotides are present in a single fragment or single piece of a DNA. Suppose this is a piece of DNA. In this DNA, many nucleotides are there. Many nucleotides are there. It is a polymer. That means many units of the nucleotides are there. And nucleotide means three things. Are you getting my point? One is a sugar. What is the name of that sugar? Deoxyribose sugar. Okay, we have already discussed about these things. Then nitrogenous base, that means purines and pyrimidines. Purine and pyrimidines. Are you getting my point? Then third is the H3PO4, which is called phosphoric acid. These three things combines to form DNA. That we have already discussed. Are you getting my point? So in this lecture, we will be discussing about the replication. How a single piece of DNA is responsible for synthesizing or to produce another piece of DNA. Are you getting my point? That is called the what? Replication. Are you getting my point? So replication is a process in which the old DNA will give rise to new piece of DNA. Are you getting my point? And this property is called autocatalytic property. See students, DNA has a specific two important properties. One is the autocatalytic property, another one is the heterocatalytic property. Autocatalytic property means the same piece of DNA or the pre-existing DNA will give rise to another piece of DNA. This property is called autocatalytic and the process is called replication. Similarly, heterocatalytic means this DNA will give rise to mRNA. When the DNA gives rise to mRNA, this process is called transcription. That we have already discussed. Okay? Transcription. And this mRNA will ultimately leads to transfer of this information from the DNA to the site where the protein synthesis will take place. That means in ribosome. So mRNA means it is a messenger RNA. Are you getting my point? That is synthesized from the DNA. And the synthesis of mRNA from the DNA is called transcription. That means the information, what is the information is present in the DNA, that is being transferred into the mRNA. Are you getting my point? And this mRNA will able to convey these informations to the site where the protein synthesis will take place. And this ultimately mRNA is responsible for protein synthesis. This process is called translation. Okay, later we will discuss about the detailed process of the translation. But one thing you must remember students, DNA will give rise to the mRNA. This process is called transcription. And mRNA is ultimately responsible for the synthesis of protein, that is called translation. And both transcription and translation they combine and termed as central dogma. What is that? Central dogma. Is that clear? So today our topic is DNA replication. That means autocatalytic property of the DNA. This is a property of the DNA, right? So one piece of DNA will give rise to another piece of DNA, and 50% of the new piece of DNA. Suppose this is the old DNA or parent DNA, you can say old DNA, and this is the new DNA piece, which arises from the old one. New DNA will give rise, will form from the old DNA. That means the new DNA has 50% of the information which is being transcribed from the old DNA, and 50%. Suppose this is 100%. Are you getting my point? 100%. So 50% of information will come from the old DNA, and 50% of information will be synthesized by this DNA only. So new DNA contains 50% of the information from the old DNA. So that this process is called semi-conservative replication. The nature of the DNA is semi-conservative. Later we will discuss about semi-conservative nature. So When DNA is responsible for synthesizing another piece of DNA, this process is called replication. And for replication, certain enzymes are responsible. These enzymes that I have written over here, right? So let us discuss one by one. These are the some important or indispensable enzymes which act simultaneously, not step by step. DNA replication is a simultaneous process. So these enzymes will be acting in a simultaneously manner, okay, to complete the process. That is called the replication. So what is the name of that enzyme? Helicase. Helicase is the first enzyme. What do you mean by helicase? Helicase is an enzyme. What is enzyme? Enzymes are protein. You must remember all enzymes are protein, but all proteins are not enzyme. You must remember this point, right? So helicase is an enzyme, right? You know that, students, DNA is a double helical structure. Are you getting my point? DNA is a double helical structure. Is that clear? And it is a coiling part. It is a coiled structure. It has 3 days and 5 days, that 5 prime, 3 prime, right? So DNA is a double helical structure, and in between these two strands, hydrogen bonds are there. Am I right? Hydrogen bonds are there. Are you getting my point? And it is a coiled structure. Helicase is an enzyme that comes and attaches to a specific part of the DNA. Are you getting my point? Helicase is an enzyme that comes and attaches to the specific part of the coiled DNA or helical DNA. Are you getting my point? This is a double helical structure, coiled structure. This enzyme helicase will come and attach to a specific site of the DNA. That specific site is called ORI site. That means origin site. Are you getting my point? So when the helicase will attach to this origin site of the or ori site of the DNA, strand, then it starts opening these strands. It starts opening. Are you getting my point? It unwinds. Are you getting my point? Double helical is like this way. So when the enzyme will come, it starts opening by breaking the hydrogen bond. Helicase especially breaks the hydrogen bond, as a result of which the two DNA strands they separate from each other. Are you getting my point? You see these two DNA strands separate from each other after attachment of this helicase enzyme. So helicase is an enzyme which is responsible for separation of these two DNA strands by breaking the hydrogen bond. So it unwinds. I am writing over here. Okay, it unwinds or opens up. Unwinds or opens up the strand. इस strand को open कर देते हैं. Are you getting my point? The next enzyme is called topoisomerase. Is that clear? After separation of these two strands, you see, with the help of helicase enzyme, these two DNA strands they separate from each other. After separation, still it is coiling. This topoisomerase is responsible for removal of tension. Tension means these coiling portions will be removed. How the coiling portions will be removed from this DNA? This topoisomerase will come and cuts the DNA in the coiling part and rotates. Are you remember? It cuts and rotate. It will rotate. It will cut. This topoisomerase will cut the coiled part of the DNA. After that, this fragmented portion will turn. As a result of which it becomes very straight. It becomes very straight, which is called the lineage. Are you getting my point? This is the role of the topoisomerase. Is that clear? Next enzyme is ligase. Ligase, what will do? Ligase simply will come and attach to the fragmented portions of the DNA, and it is responsible for attachment of this fragmented DNA into a whole piece. Are you getting my point? This is the main function of the ligase. The next enzyme is triple SP. Triple SP means single strand stabilizing protein. It brings the stability. So stability of DNA is being maintained by triple SP, single strand stabilizing protein. Okay. The next one is a primer formation, or you can say primer synthesis. Is that clear? Next one is your primase. Primase is also an enzyme. This primase, okay. So this primase is responsible for synthesis of primer, formation of primer. You can say formation of primer. Then what do you mean by primer? Primer means it is a DNA dependent RNA nucleotide. It is a basically RNA nucleotide. Is that clear? RNA nucleotide. In the beginning of my lecture, I have already taught you that what do you mean by nucleotide? Nucleotide means, see, there are two kinds of the nucleotides: RNA nucleotides and DNA nucleotides. RNA nucleotide means it is made up of three things. One is the sugar, ribose sugar, nitrogenous bases, then phosphoric acid. So let us draw the structure of the RNA nucleotide. This is the central atom, which is the oxygen. Then, okay, these are each kernel of this furanose ring that represents the carbon. And pentose sugar means it is made up of 5 carbon compounds. Okay, this is the 1, 2, 3, 4, 5. The first, okay, OH and H is there. In the second also, H, OH is there. In the third position, HOH is there. In the fourth position, only hydrogen. In the fifth position, CH2OH is there. Is that clear? This is the structure of a pentose sugar or ribose sugar. So I taught you that nucleotide means it is made up of 3 structures. Ribose sugar, this is the structure of ribose sugar. Second thing is the nitrogenous bases. Nitrogenous bases that attach to the first position of the carbon. In the first position of the carbon, the nitrogenous bases of RNA like R, A, U, C, G. Adenine, uracil, cytosine and guanine. But students, out of these 4, all 4 they do not combine to a single nucleotide. Out of 4 type of nitrogenous bases, only 1 nitrogenous base will attach to the first position of the carbon. Are you getting my point? Then in the 5th position, 1, 2, 3, 4 and 5, in the 5th position, that is in CH2OH, phosphoric acid will come and attach. Phosphoric acid means P double bond OH, then OH, then again OH. Is that clear? So now students, you see, in the 5th position, of the carbon, what OH group is there? So this OH group that makes the bond between the H and OH and it forms the water. And this kind of bond is called the phosphodiester bond. What is that? Phosphodiester bond. So again I am repeating the statement that primase is an enzyme which is responsible for synthesis of the primer. Primase, enzyme, then primer. Primase gives rise to primer. Primer means it is a RNA nucleotide. That means sugar, nitrogenous bases, then phosphoric acid. So this is the important function of this primase. Are you getting my point? The next come to another enzyme that is called the DNA polymerase 1. DNA polymerase 1 is responsible for removal of this primer. Okay? Students, you must remember what I told you, that primase is responsible for synthesis of primer. But the function of the DNA polymerase is to remove that primer. This much function have only. Right? It removes, I am writing over here, it removes the primer. Is that clear? This is the main function of the DNA polymerase 1. Right? This is the kind of an enzyme. Okay? Once again I am repeating you, all enzymes are protein. All enzymes. Whatever the enzymes are there in this universe. But all proteins that is present in our universe, they are not enzymes. Remember? So these are basically proteins. Is that clear? So the important function of the DNA polymerase 1 is to remove the primer. The next come to another enzyme that is called the DNA polymerase 3. You should not get confused between the DNA polymerase 1 and polymerase 3. Polymerase 1 is responsible for removal of the primer. However, the DNA polymerase 3 has an important role in the replication. Are you getting my point? While the DNA is replicating, DNA polymerase 3 has a significant role. What is that role? What is that role? The role is that this DNA polymerase 3 that makes the bond between two different nucleotides. See how it is, I am telling you. DNA polymerase 3 has 3 subunits. One is an alpha subunit, epsilon subunit and beta 2 subunit. Alpha unit means this subunit that makes the bond between the different nucleotides. Suppose this is a one nucleotide. Are you getting my point? And this is a second nucleotide. Okay, this is second type of nucleotide. Okay, so the alpha subunit will come and that makes the bond, that assists the bond formation, right? One nucleotide that makes the bond with another nucleotide by formation of phosphodiester. What is that? Phosphodiester. This bond is being assisted by alpha subunit. Okay. So students, the simple is, sir, you just simply told that one nucleotide attaches to another nucleotide at a given point, and by formation of a bond which is called phosphodiester bond. But how? See students. The third position of one nucleotide attaches to the fifth position of another nucleotide. Why is this so? Because third position of one nucleotide contains OH group and fifth position. Okay, fifth position of another nucleotide also contains OH group. Are you getting my point? In between these two OH group, the phosphodiester bond will form, and this alpha subunit will be responsible for that. Once again, I am repeating: DNA polymerase 3 has 3 subunits. Alpha, epsilon, beta 2. Alpha subunit is responsible for making the bond between the two nucleotides by formation of phosphodiester bond. And how the bond is formed? Because the third position of one nucleotide attaches to the fifth position of another nucleotide. Okay. Then the role of epsilon subunit. Epsilon subunit has a specific function which is called proofreading. Proofreading means it ensures whatever the nucleotide that comes in bind, whether these nucleotides are faulty or good quality of nucleotides or faulty nucleotides. That is being decided by this epsilon subunit. Okay, and beta 2 it will act just like a clamp. Okay, the next enzyme that is called the nuclease. Nuclease is an enzyme, and that nuclease is also responsible for proofreading. That nuclease is also responsible for proofreading. Is that clear? That means its nucleus also ensures whether these nucleotides are coming are faulty nucleotides or good quality of nucleotides. That is also being decided by nuclease. The last enzyme is phosphorylase. Phosphorylase means it is an enzyme which is responsible for activation of the inactivated nucleotides. See how it is happening. Okay. I am telling you well students, DNA has the nitrogenous bases like A, T, C, G. These are the 4 types of nitrogenous bases present in the DNA. Okay, adenine, thymine, cytosine, and guanine. Okay, adenine is inactivated. Why? Because adenine is having only one phosphate group, which is called monophosphate. AMP, then TMP, then CMP, then GMP. Is that clear? Now, with the help of this phosphorylase enzyme, this AMP, adenosine monophosphate, will convert into adenosine triphosphate. This TMP will convert into TTP. Then CMP will convert into CTP. Then GMP will convert into GTP. Are you getting my point? That means phosphate group, monophosphate are inactivated. These are not activated. Okay, inactive. So these inactive nitrogenous base, nucleotides will convert into triphosphate. 3 phosphate groups will attach to the adenine, thymine, cytosine, guanine, and with the help of this phosphorylase enzyme, these are the activated ones. This is the role. Are you getting my point or not? Is that clear to all? In the next video, we will be discussing how this process is happening. Thank you.